This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 572, recorded on November 1st, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dickson Despommier.
Hello, Vincent.
You sound so far away.
I am. I'm miles away.
Did you fish this week? Is that everything good?
I did, actually. Yeah, before the rains came, I went fishing. I had a great time.
Great. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's partly sunny and partly cloudy here today, but mostly sunny. And that's good because yesterday it was rainy for the trick-or-treaters. Yes. And it was, the temperature feels like 35 when they were trick-or-treating in the rain. So today it is still cold, but it's sunny.
And from Austin, Texas, Rich Condit.
Hi, everybody. It's 57 Fahrenheit here, believe it or not. That's 14 C, and it actually froze last night.
Wow.
So we skipped fall, and right. Here we are.
Exactly.
Here it's 13 Celsius and sunny, and it was raining all week. Today, now it's gorgeous out. Yeah. And we have an in-studio guest. Dickson's not in studio today.
Nope.
But I am joined. She is returning for the third time. Amy Rosenfeld, welcome back.
Hello.
Hey, Amy.
Hello.
Hi, Amy.
Hello.
I went to walk the dogs at 6 a.m. and it was dark. The sky was crystal clear. I saw Orion's belt.
Cool.
It was so neat. No clouds at all. All the stars sharp as anything.
I saw the George Washington Bridge.
That's a star. In bridges, anyway, that's a star.
I see it every day out of my window.
Yes, you do, Dickson. If you like what we do here on TWiV, consider supporting us financially. We could use your help. Microbe.tv/contribute. Couple of follow-ups before we talk with Amy about today's topic. Anthony writes, koala retroviruses are most closely related to gibbon ape leukemia virus, feline leukemia virus, and porcine endogenous retroviruses. Koala retroviruses, this was the topic of last week's TWiV, have been isolated from wild and captive koalas in Australia, as well as from koala populations held in zoos in other countries. They're members of the gamma retrovirus. Likely to be a result of a relatively recent trans-species transmission from rodents or bats. And so Anthony gives us a list of native rodents to Australia. Now, koala retrovirus phylogenetically appears to be closely related to an endogenous retrovirus of the Asian mouse Mus caroli. However, it's not on this list of rodents of Australia. So, you go figure it out.
So all of the native rodents of Australia have to be marsupials.
Why is that?
Because every mammal down there was a marsupial.
There are no mice?
Wolves, everything.
I don't know what that means, Dickson.
But what it means is that
They have a little pouch to hold their young in.
But they're mice. They're just mice.
But they are marsupial mice.
Really?
Yes.
Oh.
That's the link that I found last week said that. And this link takes us to what are called old endemics in quotes and new endemics.
Oh.
And then there's some information that several of these species are not actually endemic to the country Australia, but to the Australasia ecozone. And the headings old endemics and new endemics are used here in that sense. So whether these are all, it says they're all from the family Muridae, but I don't know if that means they're marsupials or not. But suffice it to say that Dickson is telling us that all the mammals that were native to Australia in the very beginning were marsupials. Or
monotremes.
Okay.
So this Mus caroli, it's not in Australia, but there must be something related from which the koalas got the virus because they had to have gotten it in Australia, this koala retrovirus.
Well, the hypothesis, while I was down there, I heard a lot about this stuff because obviously they talk about their indigenous flora and fauna quite often. And there was a land bridge at one point which allowed people and their animals to cross over into Australia from the archipelagos. And that's how the dingo got there as well. So it's possible that all those retroviruses could have come from, other places and then landed in Australia.
Sure. Absolutely. Kathy, can you take Steen's?
Sure. Steen writes, I enjoyed the discussion of peewee interacting RNAs on TWiV 571. I learned via a tweet from David Balcom that his paper with Andrew Hamilton describing the discovery of short interfering RNAs was published 20 years ago this week, October 29, 1999. I highly recommend a short historical overview that Sir David published earlier this year. I have always enjoyed his musing that the first RNA silencing paper may have been published as long ago as 1928, when the classical phenomenon of plant recovery from virus infection via silencing was described. Enjoy the giant virus meeting. Regards, Steen. P.S. I like Adam Loring's take on the word quasi-species back on TWiV 360 at 51 minutes 50 seconds. It's one of those terms that means different things to different people, and it becomes overly jargonized. More recently, Jesse Bloom and Julie Wolfe recorded a nice discussion of cooperation among virus variants and population memory. Cooperation and memory are in quotes. And he gives a link to the Meet the Microbiologist episode. He also gave links to the tweet, the paper, the short historical overview, and the musing. So there's lots of things in here for listeners to check out.
So this 1928 paper, of course, it may have been an effect of RNA silencing, but at the time we had no idea it was RNA silencing, right? Because we were not even thinking RNA was so important. Around that time, Wendell Stanley crystallized tobacco mosaic virus, and he said, it's an infectious protein, and it had 5% phosphate, which he ignored, and that turns out to be the RNA, of course. So that's right. I did ask Adam back then to talk about a quasi-species. Yeah, I had forgotten. Well, we have one more quasi-species letter we'll read next time from Esteban Domingo to wrap up that little arc. Now, today is an all Enterovirus D68 day, primarily because Amy just published a paper on it, and we thought we'd do a couple of snippets to sort of give a background for it. Now, you got interested in EV-68 about 5 years ago?
Yeah, about 5 years ago. It broke out in the late summer and fall of 2014. There's a lot of children developed AFM, or acute flaccid myelitis, and there was a beginning an epidemiological link between that and virus infection and respiratory disease.
So this virus has been around since 1960s, right?
Initially, it was identified in 1962 in California, but a lot of viruses apparently were identified around the same time, also in California, like Entero71.
Well, cell culture hadn't been around for virus isolation that long. I was just reading about Anders Weller and Robbins in the 50s. And so that was probably the result of that initial burst of cell culture activity. So the virus has probably been around forever. That's right? True. That's
but the discovery dates back to 1960.
Exactly. How long ago?
Identification.
Yeah.
Identification.
It would be interesting to look at archival samples and see how far back you can find it. Serologically, right?
Yes.
Yeah.
I'm not sure that we would still have the serum, but yeah. It's not something that you can do as easily as like if you were to trace polio, because it's not an oral fecal transmitted virus. So it's not going to be in the ground or in the water supply.
Yeah, but you can get serum from people. And some people have, so this 2014 outbreak, big respiratory outbreak in a small percentage of kids with paralysis, right?
Yes, like 1,200 children developed or were diagnosed with EV-68 infection. But this is hypothesized to be a significant underestimation because they're diagnosed using a biofire PCR. So you just amplify of 150 base pairs of a capsid region of the genome, but you're unable to distinguish it between a related virus like rhinovirus and enterovirus. And they're both respiratory diseases, so they just get diagnosed as entero/rhino.
All right, so this, in the news, this is called polio-like paralysis. Is that a reasonable name to give it?
If you look at the radiological scans of children who developed paralysis after this infection, the lesions in the spinal cord look very similar to polio-like lesions, but the disease is completely different.
Sure.
So polio is a lower limb disease. You really never saw quadriplegic children or adults with after polio paralysis, and as you progressed, then you had to go into the iron lung. Whereas here, children, it's an upper limb paralysis, and children actually can't hold up their head, they can't swallow, they have to have trachs put in, and a lot of them, or not a lot of them, but several of them become quadriplegic.
And this is a respiratory virus as opposed to polio, which is enteric, right?
Right. Even though it has the name.
I'm sorry, Amy, do any of those patients revert back to normal eventually?
What is normal?
Well,
like where they can walk on their own?
And they don't exhibit signs of flaccid paralysis.
No, they don't. They go through, some of them do go through a rehab where they gain some part of their limb motor function, but most of them don't recover. It's permanent.
Wow.
But what they're doing now is they're taking neurons from unaffected sides and redirecting them to the limbs that are affected. So we actually kind of monitor a little boy who just went through this, who had neurons that were redirected to his upper body parts. And now he's starting to walk again. He doesn't have a trach. And his mom just actually posted a movie of him and his brother dressed up as in tuxedos, I guess, fancy people for Halloween. And he was standing, but sometimes you do still see him in a wheelchair. But you can really,
so that's a surgical procedure, redirecting these neurons.
Yeah, it's a really invasive surgical procedure.
Wow.
And it's probably very expensive where it's not obtainable by the majority of patients. You know, you probably have to have some kind of affluence to do this, or really strong belief in the medical system that this is going to work.
So how many total kids since 2014 have had AFM in the U.S.?
550, according to the CDC, but they're still going through samples from late last year. And then there were 9 confirmed cases this year, but this year is an off year.
And globally, have there been
It's
less?
been on, nope, it's not been less. It's globally on 6 continents.
No, how many cases globally, do you know?
They don't tabulate globally.
Okay.
Globally, is it also every 2 years, and is it synced with the even numbered years like it is in the U.S.?
Yes, it is synced with the U.S. So there's some question of whether or not the virus is moving from Europe to here or here to Europe. But like usually people are more concerned with viruses that traffic from Asia because you see like flu coming from Asia. But it turns out that the enterovirus that's circulating in Asia that's related to neurological disease is Entero71, and does not have the same periodicity as EV-68, and it also causes a different disease. So it causes a demyelination.
Okay. So we have some snippets.
Can we back up just for a minute?
Yeah,
and I know we've had this discussion before, but I want to make sure everybody out there in TWiV land is on board, because enteroviruses is a big group, right?
And misnamed.
Yeah, so an enterovirus is a genus within the Picornaviridae. There are many.
There are 40 viridae, 13 species of enteroviruses. You should know this. We just published.
No, there are 40
genera of popcorn.
Include, and Entero is one of the genera.
Right, and then there are 13 species.
Enterovirus A, B, C, D, E, etc., and then some rhinos.
3 rhinos.
Right.
We just published this in Fields.
And Entero D is one, and of course polio is an Enterovirus C.
Right, but it's also it's also because it's aerosolized and there are some biophysical properties of the particle. It's also a rhinovirus polio chimera.
Yeah, but these groupings are all done on sequence.
Well, now they're all done on VP1 sequence.
Yes.
And in terms of what these viruses do when they infect people, some of them are respiratory infections, some of them are gut infections, right? And polio was
a misnomer, basically.
Mm-hmm.
Polio was a gut infection that had a small percentage of patients that experienced neuroinvasion and paralysis. Are there any other enteroviruses other than the ones that we're going to discuss that are implicated in neurologic disease?
So Coxsackie A16, Coxsackie A24 causes acute hemorrhagic conjunctivitis.
Don't some para-echoes cause?
Para-echoes are related.
CNS disease.
Yes.
Echoes also.
Well, echoes are more myocarditis. Also like Coxsackie Bs are more myocarditis.
And then you mentioned Entero A71, right?
71, yes.
70 also?
70 is acute hemorrhagic fever or conjunctivitis.
But sometimes you get CNS.
Sometimes, but very, very rarely.
Okay.
And just to complete the picture, these are all picornaviruses, so we're talking positive strand RNA virus, not enveloped, small proteinaceous capsid.
Right.
Yeah, they're all over our desk here.
Right.
They're like everywhere.
Right, but all of them that we listed, except for 68, they're all oral fecally transmitted. So the name Entero makes sense for them.
Okay, but D68 is a respiratory infection.
Yeah,
I guess we'll get more into that.
Yep.
Okay, good. Thank you.
All right, so we have 3 papers which we can snippetize.
Is that a word?
No, I just made it.
It is now.
Mm-hmm.
Yeah, and
we
here
can snip.
We can snip them first, and in these we'll lead into Amy's paper in a bit. First one published this previous March in Emerging Infectious Diseases, neutralizing antibody against Entero D68 in children and adults before the 2014 outbreak, Kansas City, Missouri, USA. Harrison, Weldon, Pahud, Jackson, Oberste, and Selvarangan. And just parenthetically, Oberste is a product of Gainesville.
Yes, he's a Bert Flanagan graduate student. Yeah.
I was on his thesis committee.
I know. We talked about him.
All right. So what's the thing here, Amy?
So the thing here is that they looked at a group of children who had a median age of 13 in Kansas City, Missouri, which was a hotbed of the outbreak in 2014. And they did this serology prior to the actual outbreak.
Banked serums.
They're banked serum. And they all test positive for the presence of neutralizing antibodies against modern day isolates and the prototype isolate, or what has been labeled as the prototype isolate, the Furman isolate from 1962. So it gives credence to this idea that the virus has been circulating under our radar for many years.
Well, this is only a couple of years before the outbreak. So it's not many.
Well, the kids range from, I think, 2 to 81.
Well, you don't know when they got infected, though.
I have no idea when they got infected because it's just one snapshot.
Oh,
Did you call someone a kid who was 81 years old?
Yeah, it was a compliment, you know.
I've got hope for the future.
You're a spring chicken there, Dickson.
So this is from 2 to 81 years. Yeah, right. From 2012 and 2013.
Right.
And so a good fraction were seropositive. And I'm really happy to say they did a neutralization assay.
Yeah, they didn't just do ELISAs.
Yeah, so they're actually looking for antibodies that block infection, right?
Right. And they block infection of the isolates that were isolated in 2014 and this older isolate from 1962. And it gives credence to this idea that there's only one serotype of the virus.
Okay. Now,
by a lot of people infected, we're talking over nearly 100%, right?
Yes.
Over 91%, yeah.
So this is a virus that, this is not a huge sample size, but this is a virus that potentially almost everybody sees.
Yes.
And so they say why this large outbreak was able to occur in a population with such high prevalence of neutralizing antibody remains unclear. What do you think, Amy? Does it remain unclear?
It does remain unclear. It's clearly probably not the virus. It's probably us who's changed. It's either the environment or genetics.
Well, they say that it may be that mucosal antibody is what's important for protection.
Right.
And they're not measuring that. This is serum antibody.
Right. And mucosal antibodies are not as effective. We see that with rhino and Gern's
they go
work.
down very quickly.
Yes.
Right. So that may be that if you measured mucosal antibody in this population, you'd have a different picture.
But you also see like that before we sanitized everything, maternal antibodies protected children for a lot longer against polio. And then when we implemented huge sanitation improvements, children weren't given maternal antibodies for as long.
Yeah, but 1960, it was pretty sanitary here in the U.S., right?
It's just an example. It's not.
Here's the thing that always, how much. So this is the question that's going to pervade this whole discussion. Why suddenly in 2014 we have this big outbreak? One possibility is maybe all of these people didn't have mucosal antibodies, but presumably that was the case since 1960. So it doesn't really explain what happened in 2014.
Right. And the virus, if you look at the sequence of the viral genome, it's very conserved, like probably less than 3% change.
Well, that might be enough.
It could be. But it depends if you're talking 3% nucleic acid or you're talking 3% amino acid. You're probably talking less than 1% amino acid.
So one possibility is that one amino acid changed and it evaded antibody.
Well, we see
but
that.
No, these are all neutralized by the old sera,
right?
Contemporaries. So that's not the explanation.
But that might not be the predominant antibody. Who knows?
Yeah.
I mean, we see that with Ian's paper and the Nature Medicine paper that the VP1 sequence that is really used, the difference is a PAL for all enteros and PSL.
One amino acid, yeah.
Right, for Entero68. So the Nature Medicine group was unable to pick up EV68 or make a signature of EV68, whereas Ian Lipkin was able to make a signature.
So speaking of Ian Lipkin, let's snippetize his recent MBio paper. Antibodies to enteroviruses in cerebrospinal fluid of patients with acute flaccid myelitis. And this was published in MBio in August. So why cerebrospinal fluid?
So the question is whether or not the neurological disease is actually due to the virus, or is it due to a para-infectious response by the immune system, or maybe it's an autoimmune response? Because you can't culture virus, you cannot culture infectious virus out from the CSF.
Of anyone?
Of children who develop AFM.
So we were talking about this morning in the polio era. Could you culture infectious poliovirus from the CSF?
Well, according to the graph that you showed me, it looks like you can, but it's kind of labeled in a very arbitrary way that is unclear.
We were reading some old 1950s textbooks, and they said cerebrospinal fluid is definitely different. There's cells in it, lymphocytes, and that's showing an immune reaction, but nobody seems to look for virus. But we do have this anecdotal story from Mark Palanch, who said that even in the heyday of polio, you could only get virus in 20% of paralyzed people.
No, you could only get viral RNA today by next generation sequencing in 20% of patients who developed polio-like paralysis.
So apparently it's hard to find viral RNAs in CSF.
Right, but I'm not interested in viral RNAs. I'm interested in infectious virus. There's a difference.
Yeah, I don't even think you could get that. Well, anyway, what do they do in this Lipkin study? They're looking for antibodies, right?
They're looking for antibodies, so they made a peptide display of the structural proteins, and they
so they take overlapping 20 mer peptides.
I think it was 12, but
and they put them on a plastic surface, and then
microchip,
and then they take cerebrospinal fluid and throw it on top, and if there are antibodies to the peptides, they will bind.
Right.
And they can be detected, right?
Right. They do both serum and CSF.
So they have CSF from
14
AFM and 5 non-AFM patients. And what do they find?
So they find that in the AFM patients, you can find antibodies against enteros, and then in 8 of those patients, or 6 of those patients in the CSF, you can find a very specific antibody to the C terminus of VP1.
Of EV-68.
Of EV-68. It's a very specific 21 amino acid sequence of the capsid protein. And you see it both in the serum and in the CSF.
This antibody. So what is the go ahead.
I'm puzzled over I don't understand the significance of antibody in the CSF. Does that mean that there had been virus in the CSF, or can the antibody get into the CSF as a result of an infection elsewhere? What's the significance of antibody in the CSF?
Well, it's indirect evidence of an infection in the CSF. But if you read Ian's discussion, he will tell you that you can get antibodies from peripheral infections in the CSF. And we know from, I think it's Jim Gurn's work or somebody, I forget. If it's not him, I forget whose work it is. But very rarely, but you do find antibodies against human rhinovirus in the CSF of patients. Mm. And we all know, and I've definitively proven that rhinovirus at least does not replicate within the mouse brain, because I've electroporated an RNA into the mouse brain and I've never detected the production of infectious virus.
So you're saying that those antibodies came from the serum into the CSF?
Yes.
And I asked Ian this, and he said, well, you can't just take CSF from anybody to do the bigger study, because they have to have paralysis or some reason why you'd want to take CSF.
Well, I don't think most people will just voluntarily let you do lumbar punctures. They're painful.
Yeah,
and dangerous.
So basically, there are antibodies to what looks like EV-D68, but you can't rule out that they did not come from the serum.
But you have to give him the benefit of the doubt, because it's clearly way above background level, because you don't see it in any other.
Yeah.
So is
this was IgG based and not IgM?
It's a snapshot of one time point.
No, IgG or IgM?
I think it's IgG.
That's a good question. I'm not sure that.
I don't think he specifies.
If IgM is there, that means that it's being produced within the cerebrospinal fluid itself.
Well, so that's nobody knows that answer.
They tested for both. It says here the samples were tested for IgM and IgG.
And they only found IgG.
I don't recall from the paper that he differentiated those.
Yeah, I'm not seeing a differentiation in the results. I think either one were positive. It was positive, but he doesn't say which was which.
If IgM is there, then it's not a breach of the blood-brain barrier. That's impossible to get across.
Really? Too big?
Yeah. So much too big.
So it must be IgG, because he says in the discussion how you can't rule it out. So that must be IgG.
Right.
Mm-hmm. Yeah.
But there are all of the components, biological components in cerebrospinal fluid. Everything is there to mount an immune response inside the nervous system. It doesn't have to leak in from outside.
No. It doesn't have to leak in. You do get an influx of pleocytosis of white cells. So initially, that's one of the characteristics that they used in the 1920s for polio, was that they saw this increase in white cell count.
Okay.
So let me read this sentence, and then we'll move to the next. Quote, we do not have access to sufficient numbers of simultaneously collected CSF and serum specimens to exclude trafficking of enterovirus antibodies from the peripheral circulation into the CNS. However, we consider this unlikely. Of 16 control patients who had antibodies to enterovirus peptides in serum, only 2 adults and 1 child also had antibodies in the CSF.
Uh-huh.
So he's saying we're probably, but he said we can't rule it out.
Right. But it's exactly what I said. It's significantly over background level, right? There's no signal to noise ratio that I discussed with somebody earlier.
So what do you think, Amy, based on this paper? Does this add to the evidence that there is EV-D68 in the CNS of these kids?
It's circumstantial evidence. It supports the epidemiology. It's not conclusive proof.
Not a smoking gun?
Not a smoking gun.
Okay.
But it would be a great tool to develop as a diagnostic, right?
Yeah.
I mean, that's what I think Ian is going after, is to be able to diagnose children. I'm not sure that you will be able to use it at the right time point, because you want to diagnose the children when they have the respiratory disease, not when they develop the CNS disease. You want to diagnose them way earlier.
All right, our third snippet is pan-viral serology implicates enteroviruses in acute flaccid myelitis. Nature Medicine, a couple of weeks ago. A lot of authors.
Came out the day before our paper. It came out last Monday.
Last Monday.
Yeah, the 21st.
All right, what did they do here? And this includes Oberste as well.
Yeah. As did the one we were just talking about.
Yeah, he's been on all of them.
Oh, he's on all three.
Yeah. Oberste and Lipkin are senior authors on that MBio paper.
Well, all samples that test positive that develop AFM have to be sent to the CDC. So they all go to Oberste.
And then he gives them out?
I don't know what he does with them. I'm waiting.
Okay, what did they do in this paper?
So they looked at a different cohort of children who developed AFM. They did metagenomic sequencing to look for
CSF, right?
CSF, yes, and serum. They did metagenomic sequencing to look for viral nucleic acid. They only identified EV-71 in one patient, but that patient had been known to be positive for EV-71, and no other pathogen. And then,
this was, by the way, 42 CSFs from AFM patients.
Right.
And only one had viral sequence by next generation sequencing.
Right.
Yeah.
But Ian addresses this also in his paper.
Yeah, he says the same thing. You can't find sequences in CSF.
It's too low.
The viral sequences are low, and there's a lot of other stuff in there.
Right. So it just goes to the bin that calls garbage.
Yeah, right. The garbage bin, the sequence garbage bin.
Yeah, that's what it goes through. But what's interesting is that bin is being reanalyzed. So for instance, Raul and Dino's lab is reanalyzing some of their garbage bins from some of their experiments, because they think that that's where they're finding these critical changes or adaptations in the genome to their selected conditions. So maybe if we went through these garbage bins, we would find them.
One person's garbage is another person's treasure. Is that what they say?
Yeah, that's why you're supposed to
Amy, is there a plaque assay for this virus?
Yes, I do plaque assays all the time, Dickson.
That's what I thought. That was a leading question. So why can't you find the virus doing the plaque assay? Even if you had one particle, is that enough to start a plaque?
I would think so, but...
have you ever tried it with cerebrospinal fluid?
I don't have these samples, Dickson. They're not given out.
I see.
They're not giving them out. You don't get a lot in the CSF puncture, right? You get a mil at the most.
I've never done one. I've never had one done. I have no idea. But they don't give them out. I think, I don't know anybody, although maybe Kevin does.
Kevin Messakar?
Yeah, maybe he's giving out some.
So no sequences, but they did find something else here, right?
They did find the antibodies to enteras that were enriched. They did this by phage display of the entire polypeptide they cleaved up.
Of the virus. This is called Veer Scan.
Yes.
Which we talked about on TWiV before. Mm-hmm. You have phages that each produce a piece of the viral genome polyprotein, and then you can see which ones bind your antibody, right? And then when you sequence the phage genome to find the epitope, right?
Mm-hmm. Yes. And so they use the same peptide. They identified the same VP1 peptide that is pan-enter that Ian used, but clearly states you cannot identify EV-68 with because of the one amino acid change, the A to the S. And then
which genome did they use for the phage display?
I think Coxsackie.
I see. Why didn't they use Entero68?
I don't know.
Okay.
And then they found an antibody to the RNA-dependent RNA polymerase
in the CSF.
In the CSF.
How many kids?
I forget. Not a high percentage, but they did find it.
So this paper says enterovirus is not EV-D68.
Right.
Because their peptides cannot pull out a EV-D68 antibody.
Right.
Whereas Ian's, he got a antibody to a EV-68.
Well, he made
peptide.
a specific fragment.
Right, because he used EV-D68.
Right.
And the difference, as you said, is one amino acid.
Right. For that specific N-terminal part of VP1.
So I thought it was curious that this paper got lots of press coverage, front page news, but it's less definitive than the Lipkin study. And I asked him why his didn't get more press, and he shrugged.
Yeah.
This one's in Nature, the other's in MBio.
You think that's it?
Mm-hmm.
Sure. The people who write well, I also don't know the extent to which the authors might have promoted a press release in either case. That's a possibility.
Yes, that is. I heard him mentioning the press release. Yeah.
I think he also had one. His paper was also picked up by the NIH and publicized. I got emails from the NIH about it. I know it's been discussed in our EV-68 working group on Monday, the same day that this pan-entero paper came out. So
they say in the discussion that these results do not fulfill Koch's postulates for causality.
Right. It's one time point. Doesn't say anything about convalescence, doesn't say anything about conversion from IgG to IgM.
IgM to IgG.
Doesn't say anything. It just says that there's a potential antibody. It doesn't correlate. They even go even further. The presence of the antibody doesn't correlate with an increase in white cell count in the CSF.
They do mention the Bradford Hill criteria, which we had run into way back when. Was that with Zika? Or was that... Do you remember that, Vincent? Or Rich?
No,
yeah, the Bradford Hill were sort of an alternate criteria relative to Koch's postulates.
But I think that they state that it doesn't fulfill that criteria. It's getting closer, but it doesn't fulfill that criteria.
Helping to fulfill,
Right.
Yeah.
I think what is great about this is that the parents whose children have developed AFM after EV-68 infection are really active, and they're really aggressive in trying to promote research and understanding what's causing this disease. And so I think we'll get there, but we're not quite there yet.
Before we move to Amy's paper, I want to point out two things about these two papers about the CSF one, and they both mention the fact that they don't have the best controls optimally matched with respect to age, year, or season of collection, and that they need to do prospective studies with appropriately matched controls from the same outbreak season. So that's a limitation they both have. And then in this, particularly in this Nature Medicine paper, when they were talking about the fact that the whole VP1 ELISA couldn't consistently identify specific individual EV types, probably they say owing to cross-reactive immune responses to conserved linear EV antigens. They say future studies using well-folded virus rather than linear phage display peptides or individual viral proteins may be more fruitful. And so I had just never heard the phrase well-folded virus before.
I love it.
So,
well, I think when people did these studies, we were still on the fence about this every other year And there's really, if you take all 3 outbreaks and you add in the number of cases, it's 550. It's not a lot of cases. And the last one was the greatest one at 225.
That's 2018.
2018. So we're just trying to put in place, or trying to get the NIH and the CDC to put in place where you can actually do a prospective study, because 2020 is coming up, and that's the on year.
So could you elaborate on this periodicity? It's the virus infections themselves, or the acute flaccid myelitis that happens apparently peaks every couple of years, and which and why is it?
So it's both. So virus infection peaks every other year in the late summer and early fall, just like polio does. And AFM correlates slightly. There's a slight delay because you don't automatically develop AFM when you get infected.
And is there, what's the thinking about that periodicity every other year? Why would that be?
It's unclear. We have lots of thoughts. So some people think it's virus evolution, but, we're virologists. I can't accept that. It's not a good explanation. Others think maybe it's a naive population. And that's when you see it, because the naive population is of enough numbers that you'll see it above background, because there's always a small amount of non-infectious AFM.
Mm-hmm.
So enteroviruses are typically periodic in their epidemiology, and you wait to get a cohort of immunologically naive people, and you have an outbreak.
Right, but
Mm-hmm.
right.
Same thing with other viruses too, right?
Right.
Mm-hmm. Flavies, 5 to 7 years.
Yeah.
So that's probably it. I mean, the real question is, why did we suddenly in 2014 see this huge outbreak that we'd never seen before?
Especially when you look at that, it's not just from the first paper that we discussed. You see that in Denmark and in Finland, a significant percentage of the population that is now 30 or above are seropositive for Enterovirus D68 antibodies.
Now, at the meeting in Copenhagen where Ken Tyler talked about this, someone asked him that question. They said, why did we have this big outbreak in 2014 and subsequent ones? And we'd never seen this before. And he said, he didn't know the answer, but one possibility is that a change in the virus occurred that made it transmit better among people by aerosol. So you got more infections.
Doesn't make any sense either.
Now, what do you think of that? You say there's not enough changes to do that, but you only need one amino acid change.
Right. I don't think that's the...
Every other year? Come on.
I don't think that's
No,
correct.
not every other year. Just that first 2014.
I don't know if that's correct.
So if you look at 2014 virus versus 1962, there are a couple of amino acid changes, more than a couple, right?
I've aligned the whole genomes of all of the available full-length genomes. Maybe there's approximately 1% of the amino acid changes, 6% of the nucleotide, just based on the fact that the code is degenerate.
But you could never prove that any of those enhance transmission.
Well, first we have to establish an animal model, and then we can prove some stuff.
If you have a transmission animal model, right?
Well,
but even then, one will say it's a mouse or a ferret or whatever it is that you can
Right, it just gives you some insight. Yeah. But like, for instance, you don't see the seasonality of polio transmission as you move closer to the equator. Mark Palanch and Oberste did that study last year and published it in PNAS. So the more humid the climate, the more temporal the climate, the less the periodicity.
I'm not talking about the 2-year periodicity here. I'm just talking about the sudden appearance in 2014.
Yeah, I know.
Now, back in 1900, there was a sudden appearance of polio outbreaks, which we ascribed to improved sanitation. However, as I said before, in 1960, there was no change in the sanitation.
I don't know. Maybe we've screwed up the climate so much. Maybe this is proof of climate change. I don't know.
Arg.
Well, I mean, it would, except for the fact it doesn't have a vector, right? You could argue that we see West Nile and other things that have an insect factor due in novel places due to climate change, right? Because
those are vector-borne.
Well, that's what I'm saying.
Yeah. But, so what other periodicity events that occur in nature that has nothing to do with this infection, supposedly, but it might, do we have a diurnal periodicity for, let's say, sunspots? No. Climate change? No. And you start looking around for the, maybe the magnetic fields of the Earth switch back and forth or something crazy. It's got to be something other than what that virus is doing, it sounds like.
Dickson, the diurnal periodicity is antibody levels in the population. Most likely. I would buy that. But what's the puzzle is the first 2014 outbreak, why that happened. It's very hard to explain.
Yeah, but you have
can't ask why questions,
right?
Well, yeah, we'll phrase it without a why.
How?
So I'm missing something here because the first paper that we discussed that was talking about the 2014 outbreak or trying to explain it did serum samples from way before that and found that the vast majority of individuals have antibody to this virus. So I don't understand. So the virus has been around and is very prevalent and presumably has been around for a long time. So what constitutes an outbreak? When you're talking about an outbreak, you're talking about a bunch of people who present with symptoms and then wind up getting diagnosed with this. I don't understand what constitutes an outbreak over this background of so many people getting infected. Is it a cluster in time? I don't get it. So
I think what the confusing thing is that what are the symptoms that you're using to define the outbreak? Mm-hmm. And the symptoms that we're using to define the outbreak is the increase in children who developed paralysis.
Okay.
We're not using the symptoms of, are you sniffling? Are you having cold symptoms? Are you coughing? Because the majority of those patients don't go seek medical attention.
Right. But this outbreak was severe respiratory disease in kids. That's what triggered it first. And then subsequently they had the associated paralysis, right?
So but you always see you always see children who have severe respiratory disease.
But not in the numbers at the end of the summer like this.
But it wasn't the severe respiratory disease per se.
But
it was the severe respiratory disease, and then you came back 10 days later and you said, my child is paralyzed on the left side, can't lift up their left arm.
I'm not aware of any late summer respiratory disease outbreaks before. I know in the winter you get rhino outbreaks, you get flu outbreaks, but maybe in other countries there are enterovirus respiratory outbreaks. But we hadn't seen them in the U.S.
Right, but you're also lumping children who come in with severe respiratory disease with asthma and other chronic autoimmune pulmonary diseases. Yeah. And a lot of these children who had severe respiratory disease with EV-68 are asthmatic.
Okay. All right, so let's move.
Before we move on.
Yes.
And the people have maybe already written letters. When you guys are talking about diurnal, I'm being pedantic here, but that means daily.
Yeah,
I'm thinking that you meant biennial.
Yeah, I'm sorry. I just repeated what Dickson said. Totally. I figured he was right, but I should have known better.
No, he was wrong.
Sorry, I'm sorry.
No, I thought about that just a moment after I said it.
It's okay. I read papers
biannual.
I read papers that call it
with an E.
Right. I read papers that call it biannual, where it sounds like it's every half year.
Right. That would be with the A. Biennial.
Biennial.
We're talking
right. You're talking BIE. Yeah.
Right.
So let's talk about your paper now, which you published a week ago in Bio. Neurotropism of Enterovirus D68 isolates is independent of sialic acid and is not a recently acquired phenotype. Rosenfeld, Warren, Racaniello.
And so before we jump into that, could we summarize what we've learned from these snippets to set up this paper? I can't do it. I'm asking you or Amy to do it.
So what the snippets say is that they enhance the circumstantial epidemiological evidence that EV-68 is today's etiological agent of AFM.
Okay.
And that is critical because we all thought that acute flaccid myelitis would disappear with the eradication of polio. And that obviously was an underestimation.
So I should just throw out that this AFM outbreak is not because we are no longer immunizing against polio.
No, but somebody at a job interview did ask me that.
Right, because we're still immunizing against polio.
Well, they asked me if it was, yeah.
So they asked you, what did they ask you?
They asked me if it was due to the development of antibodies because we've changed from OPV to IPV.
Oh, I see.
And whether or not the antibodies were against polio were acerbating, more like antibody-dependent enhancement of disease. But actually, antibodies against polio don't cross-react with EV-68.
Right. So,
and you don't think it's anything that, because there's no paralytic polio anymore, that now our attention is turned to the next kind of flaccid paralysis, and that's this? Because of the sudden onset in 2014. Like, if that were the case, it should have happened longer ago than 2014.
Yes. Plus, if we were going to just use neurological disease as saying anything that related to polio or symptoms related to polio, then we should have focused in on EV-71, which has been epidemic throughout the Far East since the early 2000s, maybe the late 1990s.
That causes paralysis.
That does, in some cases, does cause paralysis. It's slightly different, but it does cause paralysis.
That's a good question because this AFP, acute flaccid paralysis, was defined by WHO for looking for kids with polio when it's rare. After much of polio was eradicated, they said we still have to find the rare cases of polio. So they said we'll do an AFP surveillance looking for kids with acute paralysis, and then take stool and see if they have polio virus in the stool. And if they do, then we say it's polio.
Right. That's much easier than something that's respiratory.
So the curious thing here is that the kids develop respiratory disease, and then a few days later, as they seem to be recovering, they develop flaccid limb paralysis, right?
Yeah, there's a window. It's, I think, average is 10 days after you initially are diagnosed.
And that's, so one of the another puzzling aspect of this infection is that no one has found a virus in the blood. And, if the virus gets into the respiratory tract and is multiplying there, the idea would be then it spreads to the central nervous system via the blood, like measles virus does, right?
Right.
You can find measles in the blood, but you can't find EV-D68 in the blood.
Right. But you can very rarely find rhinovirus in the blood. I believe in fields by Gerns, you can only find it in immune-compromised individuals. This is a respiratory disease. One has to keep in mind that the respiratory tract is a passive conduit. It's not analogous to the gut.
But measles starts as a respiratory.
No, I understand that.
In fact, there's coughing and sneezing to spread it to other kids,
but I believe it had to go into the blood first to get to the respiratory tract because it infects the cell. It
enters the respiratory. Yes, to get out again. You're right.
Because it enters the cell of the respiratory tract at the basolateral surface. So it's on the bottom that is towards the blood. This is entering on the apical surface, so it's actually outside the body.
Do you think there is a viremia in EV-D68 infections?
I think it's very transient, and we're not sampling at the right time.
Okay.
And I'm not sure that we will ever be able to find it because we sample when the children develop severe respiratory disease. If you make that analogy with rhino, that's way past peak viremia, virus replication.
Does a virus have to traffic through the blood to get to the nervous system?
So that's the question. Because there's only one skeletal muscle in the respiratory tract, and that's the diaphragm, and it's separated. It's physically separated from the respiratory tract, the passive conduit.
There's no innervation in the.
There is there's smooth muscle, and then there's, I'm going to get this wrong, parasympathetic, where you don't have where you don't have control over your nervous system. It samples, it takes the pH, and then it triggers a response to the nervous system. Plus, those neurons are in the wrong portion of the brain.
Yeah, that's what Adam Loring told me once. I said it could spread by neural pathways from the lung to the brain. He said, no, those are not the motor centers that go from the lung.
Right.
Yeah.
So what's interesting is that the children who develop AFM have droopy face, droopy eyelids. Those cranial nerves are much further back in the brain. So maybe it's entering, maybe we have it all wrong. Maybe just because we do the diagnosis from a nasopharyngeal swab, that's not the actual initial site of infection. But, we don't really need to know what cells it infects.
Can you get to the brain through the nose?
Yes.
Yeah, the olfactory bulb, yes, you could.
Right.
That's in the front also.
Yeah.
And why do you really want to discuss Flexner?
That's funny. So Amy's referring to Simon Flexner, who thought that polio entered the olfactory bulb.
And that you could use nitrous oxide, the white stuff, on your nose and prevent it.
Well, not nitrous oxide.
Well, what's the cream that you put on your nose so you don't get a sunburn?
Zinc.
Zinc
oxide.
Zinc oxide.
Whatever. I knew it was this white cream. You see people on the beach with it.
People use nitrous oxide for other things.
Yes, they do.
Isn't that the gas that the dentist gives you?
Yeah.
Yeah,
right.
laughing gas.
Yeah.
But it's also a recreational drug.
How would you know that, Dickson?
I've seen movies about it.
Oh, yeah, sure. I think you have a tank in your home.
No, I don't.
That's why he couldn't make it in today.
So, Amy, tell us what you were trying to do in this paper.
So we started our studies to, one, identify the cell receptor of the virus to understand how it enters and do a lot of biochemical and biophysical properties studies about viral interactions with the receptor and entry and encoding and antibodies, as well as whether or not the virus is actually neurotropic or not. And if the isolates from the early time points when we first isolated or identified the virus to today were actually different.
Okay.
So
tell us about the receptor. What did you do?
So there's 4 molecules that have been defined as the receptor, and this we only reported on 1 or 2 of them. The 4th one was recently identified, but it's a cell adaptation. So it occurred while this virus was adapting to cell culture. So I'm not clear that that's actually biologically relevant, because we know that picornas do adapt to their environment in cell culture.
So out of curiosity,
that's heparin sulfate.
Okay. So that's pretty typical.
Right.
For viruses to adapt by learning to bind to heparin sulfate.
Yeah. So like it's well documented for foot and mouth disease, which is a related picornavirus. And Palmenberg has recently reported this for rhinovirus C. We had eliminated decay accelerating factor, which is a co-receptor or receptor for Enterovirus 70. We're just not reporting it because when they described the interaction or the potential of it being the cell receptor for EV-68, it was just one sentence in a paper, in an obscure paper with no data supporting it. But the real molecule that has gotten a lot of publicity as the cell receptor are 2 molecules. They're ICAM5, which sits in the telencephalin region, which is right at the front of the brain. It's not found on motor neurons. It's not found within the respiratory tract. It's not found on any of our cells that are infected with EV-68. And others have also demonstrated that if you remove it, there's no effect on virus replication. And they also have reported that it's not found on their motor neurons, their motor neuronal cultures. And so the last molecule is sialic acid. And sialic acid, as you know, is a 9-carbon sugar, and it's very common on proteins that are found on the cell surface. And it has been reported to be the receptor, and now it's no longer the receptor, for Coxsackie A21. And it's also a co-receptor or an attachment molecule for other picornas. So we wanted to know whether or not that mediated virus entry. And so we removed it from the surface of the cell using neuraminidase, which is an enzyme that cleaves sialic acid, and found that, yes, there's a delay for some isolates, but they all replicated and produced infectious virus.
What kind of cells did you look at?
So we looked at multiple cells. The paper just covers 2. It covers RD cells, which are human rhabdosarcoma cell line that that is traditionally used to propagate and plaque the virus, and also murine N2As, which is a neuroblastoma cell line. But we looked at human neuroblastoma cell lines. We looked at human lung cell lines like A549. We have immortal cell lines that are not transformed, that are also lung, and it had no effect.
So there is a structure of Entero-68 by Rossman, which is made with sialic acid, and it binds in a pocket, and he says that that's evidence that it's a receptor. So what do you think of that?
He did the structure. What he did was he crystallized the virus, and then he soaked the crystals into a solution of sialic acid. Mm-hmm. It's not the same as co-crystallizing the complex. Mm-hmm. And it does not cause the conformational change. So this is analogous to what we knew about Coxsackie A21, that it bound sialic acid, but it never caused the conformational change.
Conformational change leading to release of the RNA, right?
Release and uncoating of the RNA.
Mm-hmm. And I think you told me once it's just sitting in some holes in the particle.
It's sitting in the pocket. And
so it actually sits in the pocket below the receptor binding canyon, right?
Right. And in some cases, it looks like it excludes pocket factor in some of his crystals.
So this virus, where you soak them in sialic acid, can you infect cells with those?
He did not look at infectivity, but being trained by you, I did, and really interested in this. I did the classical experiment that you had done with soluble poliovirus receptor. I incubated virus in the presence of sialic acid, increasing concentrations of sialic acid, both at 4 and at 33, and then plaqued
sialic acid or neuraminidase?
Sialic acid. Because if it stimulated the conformational change, I should lose infectivity at the particle.
Right.
And my plaques and I never saw a decrease in infectivity because I plaqued it all out.
So if you do that with soluble polio receptor, you block infectivity.
Right.
Yeah.
So I did all of those classical experiments, a lot of time in the cold room. And there was never a loss of infectivity.
Okay. So others say that sialic acid is participating in entry. What's your take?
So I think it participates, but I think it's more similar to where it enhances receptor interaction. But none of these molecules are the receptor.
Okay.
So sialic acid, the virus could bind to sialic acid, and that makes it hang around on the surface of the cell a little more, but it's really waiting for the real receptor.
Yes.
Okay.
What about your result with the red blood cells, where you say no isolate agglutinated human red blood cells?
So you can also look at the interaction classically by hemagglutination assays, where you mix serial twofold diluted red blood cells with a stable concentration of virus and ask whether or not the red blood cells pellet. And so if you look at human red blood cells, in every case, the red blood cells pelleted, so the virus did not bind them. However, aren't you alluding to the fact that this is an interaction between sialic acid and the isolates that isn't happening then? Right.
In this case,
it's not happening with red blood cells, with human red blood cells. However, if you look at guinea pig red blood cells, which is what was used to define the interaction, you do see that
sialic acid. Okay.
Some isolates agglutinate those cells, so they prevent precipitation of those cells. But it's not like all the isolates from 1962, the Furman and the Rhine strain did it, and all the isolates from 2014 didn't do it. They all have differences. So some of them do and some of them don't.
And then I'm assuming that the guinea pig sialic acid has different linkage, branching, whatever, from what's on the human red blood cells.
Yes, it's somewhat different. But there's also differences in the respiratory tract, too. Like 2.3, I think, is more lower, and 2.6 is more upper. And
what do red blood cells have? It must be 2.6, right?
2.6. But, I mean, as I said, we could be looking at completely the wrong compartment, right? I mean, for hundreds of years, we thought that flu infected at a certain compartment. And then the lady at the NIH showed that it infects at the soft palate, which has like a huge amount of sialic acid in a very small area. I think it's increased like 1,000-fold compared to what's on the surface of the respiratory tract. Do you remember, she had the forehead?
Yeah, I remember that.
She got the head. Do you want some soft palate?
I don't want the heads.
What was her name?
She's in Australia now.
Yeah, she moved.
Is it Kanta?
Kanta.
Yeah.
Kanta Subaru, yeah.
Yeah.
She told the story.
Yeah. She was a speaker at ASV.
Oh, she told it at ASV, yeah.
Yeah.
She talked about the heads.
Yeah.
I can't
She had the four heads
I
come
know,
to her lap.
She asked for pallets, she got the whole head. That's horrible. It's horrible. I mean, having just been in Galveston, Rich, where we saw heads in jars, man, I can't imagine getting them in a box. All right, so that's sialic acid story, and as you said earlier, you're interested in finding the receptor for this virus, which you'll probably do in your own lab one day, right?
Hopefully, yes.
Okay.
Yeah.
And now, what's the second story is about infecting neural cells. Tell us about that.
We wanted to know whether or not the virus is neurotropic, the ability to infect cells of the central nervous system. So we looked at it in two different ways. We took human-induced pluripotent stem cells and terminally differentiated them into neurons or astrocytes and then infected them with our multiple isolates of EV-68 and observed that all of our isolates were able to infect astrocytes, which was very surprising.
Why?
Because AFM or paralysis is thought to be a neuronal death thing. And so you wouldn't have expected that this cells that maintain the neurons would be the target.
But they also infected neurons. Right?
Yes. 6 of the 7 isolates infected neurons.
And the key here is that the earliest isolates you have are from 1960s, right?
Right.
And so this addresses
the evolution question.
Yeah, many people say in reviews that the virus has changed to be able to infect nerve cells, but this is doesn't seem to be the case, right? At least as far as neurotropism goes, right?
Right. Neuroinvasion is a separate issue. You can only do that in an animal model.
And we don't have an animal model yet.
Not at the moment.
Although
I was just going to say, maybe you could just describe what you mean. What's the difference between neuroinvasion and neurotropism?
So neuroinvasion is where the primary site of infection is outside the central nervous system and you have to enter the central nervous system.
So like respiratory tract.
Right. So like from the gut, you go through the blood and then you enter the CNS from the blood.
You could either enter from the blood or from nerves. Either way, that's neuroinvasion, right?
And since there's no skeletal muscle in this part of the respiratory tract, the idea that you are going through the neuromuscular junction like you see from polio doesn't really hold.
So do you think that the virus could have evolved to become neuroinvasive?
I think that the virus could have, it's not specifically evolving to become neuroinvasive. It's evolving to escape the innate immune response.
Mm-hmm. And that could have led
to neuroinvasion.
Okay.
Right? Because now it's going to cause more damage. You're going to be able to spread easier.
Mm-hmm.
And that is what allows you to become neuroinvasive.
There are genetic differences. I'm just checking. There are genetic differences between the earlier isolates and the isolates that are circulating now.
There's approximately 1% amino acid change.
Okay.
And okay, so now we've talked about what neuroinvasion is, and I just want you to then describe what you mean by neurotropism.
So can it replicate and produce infectious virus in a cell that is from the central nervous system? So either an astrocyte or a neuron.
Okay.
Doesn't imply anything about killing the cell, right?
No.
Just replicating.
I'm just using it as
Actually,
it produce
reproduction, I would
right.
say.
It's reproduction. It's just can I infect and make infectious progeny?
Okay.
Okay. And neuroinvasion is, can I get there from somewhere else?
Right.
Which you did not look at in this paper.
I did not
right?
look in this paper because it's been very hard to establish a small animal model for the virus. The virus has a very broad spectrum of pathologies that you would have to mimic all at once.
So there has been an animal model published where they inject it into the muscle.
There has been a small animal model where you do multiple different types of inoculation into very young, maybe day one old pups.
Mm-hmm.
But you can take a neuroattenuated poliovirus vaccine strain and inject it into those same age mice, and those mice will develop paralysis
because they have no innate immunity, right?
Or very little innate immunity.
Yeah.
And so I'm not sure that is I would say that is not an immune compromised animal model. I want an immune competent animal model.
Right. Okay, so you said all these isolates replicate in astrocytes and neurons, right, of human origin.
Right. One did not replicate in human neurons, but all of them replicated in astrocytes derived from human-induced pluripotent stem cells.
And the one that didn't was from a 2014 outbreak.
Mm-hmm.
What do you make of that? This is a virus that was isolated from a person, right?
Right, but none of the children had acute flaccid paralysis. So all of the isolates that have been released by the CDC are of children who developed respiratory disease only.
But I understand they're now going to release some from kids with AFM, right?
Right. I'm waiting.
Are you waiting?
I'm waiting. I'm waiting with bated breath. I check every day, maybe multiple times a day, just in case I missed it.
So these are induced pluripotent stem cells. Where do you get these cells?
So we purchased them.
Okay.
Initially from a company that is a collaborator or has a collaboration with the stem cell bank at the University of Wisconsin.
And you do the induction in your lab?
They're terminally differentiated, but you finalize them when you thaw them, and yeah.
Okay, so some of the process is done by the supplier.
The majority of it is done by the supplier for this experiment.
What's your feeling for how closely these mimic the real cell type in the animal?
Well, they have a very similar transcriptomic profile, but there's a lot more that is required in an intact animal. Like you need to have the proper conductivity and electrophysiology. They kind of make synaptic connections or what looks like synaptic connections with each other. But it's, there's a lot of extracellular stimuli that are secreted that maintain the neuron by the astrocyte when you're in a brain. And since this is a purified neuronal culture or a purified astrocyte culture, those extracellular cues are missing.
Okay.
You also did some work in mouse cells, right?
Yes.
Tell us about that.
So I did some induced pluripotent mouse cells, and but really what I've taken advantage of is the organotypic brain slice culture that we developed when we were studying Zika.
Zika by the slice.
Zika by the slice. Would you like pepperoni with that?
You do like pepperoni, yeah. That was your last TWiV, right? No, that was before that. It was your first TWiV. The second one was in Dino. Yeah,
was with the Raoul.
So tell us how you make these brain slices.
So you generate brain slice cultures from postnatal day 2, 4, and 10 mice. Mm-hmm. So you have a gradient, and the reason why this gradient is important is because when you looked at the model that a group in Colorado defined, they see a loss of infectivity at approximately day 8. So they are unable to infect mice after a certain age. And so we looked at, and you want to know whether or not as the brain develops and becomes more terminally differentiated and more adult-like, if the virus is able to cause to infect cells, because you see the paralysis at a very specific age.
Mm-hmm.
Those people working on the animal model before, how were they infecting the mice?
So they did a variety of non-physiological and physiologically relevant routes of inoculation. So they did IN and IC. I guess you can make IC physiologically relevant. And very few mice, I think 2 of 76 mice after IN inoculation developed paralysis.
These are
That's intranasal.
Yes.
1 day old mice?
1 day old mice.
Okay.
And then, but the mice that developed paralysis most efficiently were the mice that were intermuscularly injected.
Okay.
I think that's probably because the virus is replicating and you're getting higher titers out of the muscle?
I don't know. The muscle wasn't really plaqued.
I see.
And it was never shown that when you developed paralysis, that was at the peak of viral reproduction in the muscle, just like within polio. Plus, they didn't cut the sciatic nerve. So I don't know.
So going back to our previous discussion, you also don't know in those cases where whether you're talking about neurotropism or what was it? Neurotransmission?
Neuroinvasion.
Neuroinvasion.
Yeah.
Doesn't differentiate those.
No. It's just reported as the mouse developed paralysis.
Okay.
So in your slices, what do you see?
So in my slices, I say that all of our isolates are able to replicate efficiently. There's the production of infectious virus progeny and released into the supernatant of all the cultures. Mm-hmm. And by indirect immunofluorescence, I can identify sites of viral infection, the neurons and astrocytes. And then to really clearly look at astrocytes, I purified astrocytes from the mouse brain at an early time point.
Mm-hmm.
So in the brain slices, neuroinvasion is not an issue, right?
Right.
You're looking just at neurotropism.
Right. You inoculate the virus directly onto the cells. Onto the cells?
Mm-hmm. Yeah,
onto the cells.
They get in on their own.
Yeah.
Because our lab made mouse brain astrocytes from mouse brains, I want to hear a little bit about your protocol for doing it. What was involved?
So I used a combination. Well, actually, it was just confirmed. So Richard Valley's lab does neuronal cultures, and then Glenn Rawl also does this. So you approximately between day 1 and day 4, you isolate the brain. You remove the olfactory bulbs. You remove the brainstem. You remove the hippocampus from the bottom of the brain, and then you remove the meninges.
You're doing this on a day 1 old mouse, right?
Well, yesterday I did, but other times I've done P2s or P4s.
So this is not that you do this under a dissecting microscope or something?
Well, to remove the meninges, yes.
Yeah.
You do it
because this is not a big thing, right?
No, it's very small.
Right.
Okay, so this is a little bit different from how we made our astrocytes. We always made them from day 1. But we didn't do all the dissections that you described. And then I can't remember, I meant to look it up. I can't remember if there was a gradient step in there of something like on a Percol or something. I'm conflating it with endothelial cell preps. But anyway, then we cultured them for 2 weeks, and then we did this shake in the cold. Is that part of your protocol?
No. So what we do is, it depends on what I'm looking at, but so we place the hemispheres after the meninges has been removed in trypsin to make a single cell culture. Mm-hmm. And then you place it basically in DMEM, regular tissue media, culture media with 10% heat inactivated fetal bovine serum, plus or minus pen strep, depends on who you talk to. And then if you want to do IF, then you make sure your slips are pre-coated and really well washed from the polylysine, because that's toxic. And then what happens is all the motor neurons, since all the neurons are going to die because you're not selecting them. So you're not adding like B27 or neural growth factor or any of that. And so these cultures look pretty crappy for the first 3 days. And then you start to see the astrocytes. And you can probably, depending on what size dish you place this in, get a confluent monolayer in 7 to 10 days.
Do they actually divide, or are they just
no, they do divide.
Survive? Okay.
No, they do. You can see them dividing. And then if you stain with GFA and antibody against GFAP, which is the signature intermediate filament of astrocytes, your culture is pure astrocytes.
Okay. So our protocol, I take it back, we did it day 1 or day 2, and we did isolate the cortexes, trypsinize them, triturate them, and plated them with macrophage colony stimulating factor and granulocyte macrophage colony stimulating factor. But then this was the thing, in 2 weeks later, then we shook them in the cold, and microglia shook off and the astrocytes stayed stuck. So it was never, it was more of an enrichment. So I was curious if you had majors that way, but obviously you didn't. And then the other question is that you said that you incubated them at 33 degrees. And why is that?
Well, I incubate them at 33 degrees after I infect them, because the virus likes to be at 33.
Okay. I never knew that. Okay.
Yeah. So a lot of respiratory viruses seem to prefer 33 degrees. But I think actually William Jackson's lab that works on EV-68 grows everything at 37. But don't hold me to that. But when I'm initially isolating the astrocytes, everything's at 37.
Mm-hmm. And you have this culture and you say it's pure, but did you ever do immunofluorescence looking for virus in those cells? I'm just wondering, do you get all of them infected, or you haven't looked? So
we've looked. It's a bit difficult. We haven't really been that successful, but I know the cultures are all pure astrocytes because I have a uninfected well that I look at, and I stain for both GFAP, IBA1, which is a microglial specific marker. It's a calcium phosphate. It's a calcium protein. And I counterstain for NISL, and I never see positive signals for NISL or IBA1. I only see GFAP in these beautiful filaments.
Okay.
And we have been trying to look at virus infected. I just haven't gotten the timing right.
Okay.
So what are you infecting adenoviruses onto these things, Kathy?
Oh yeah, we were looking at mouse adenovirus infection of them.
Do they infect astrocytes? Does that virus? Yeah, cool.
Mm-hmm.
All right, and then one more experiment in this paper. It's cool.
Yes. So to look at whether or not the innate immune response has a role, and if we've kind of avoided all of this by using explant cultures, we looked at 4-week-old wild type black 6 C57 black 6 mice and mice that lacked the alpha beta interferon receptor. And we did intracranial injections and found that if you remove the innate immune response, all of the mice succumb except for one to paralysis. And we used 4 different isolates. So an isolate that the Lipkin lab isolated in 2009, 2 isolates from 2014, and the Fermon isolate from 1962. And so this falls well in line with what we know about other picornaviruses, where their ability to cause paralysis or cause neurotropic disease is linked to the innate immune response.
Do you think kids who develop AFM after EV-D68 have defects in their innate immune genetics?
I wouldn't be surprised if we found out that they had SNPs in some component of the innate immune response. So it's been well described by the Casanova lab that if you have a point mutation or an amino acid change within the MDA5 protein, that those children are unable to resolve concurrent entero/rhinovirus infections, but they're fully able to resolve flu infections and other infections plus bacterial infections. So obviously, from a knockout mouse, the subtleties of the human genome or genetics is not really observed.
Yeah. But if one day you could identify SNPs in these kids, you could put them in a mouse model and test them, right?
That's one way to do it. The other way to do it is identify SNPs in the mouse and ask
and
whether...
put them in people?
No, ask whether or not...
Just kidding.
Ask whether or not those children have
sure.
orthologs that have defects.
Do you think we should make a vaccine against this virus?
I think that it'll be really hard to make a vaccine against this virus. There's a lot of enteros throughout. So what are you going to do? Make a vaccine against every entero?
No, just this one.
But if you made it against every entero, would that be bad?
Oh, that's a good point, yeah.
Well, that's a lot of vaccination of children.
Well, you can make it all in one shot.
No, you can't, because you can clearly see that, like, just for polio, monovalent IPV or OPV is way more effective than trivalent. Isn't that why we give 3 vaccines and then 2 boosters?
That's right.
Yeah, but what about some other, like for FMDV, is that a multivalent? Other picornas maybe for which there's vaccines. It doesn't have to be that way.
Well, let's say you could and make a vaccine against 20 enteroviruses. Is that viable? Because I think EV-68 alone is not compelling. I know the parents whose kids are paralyzed would say they wish they had a vaccine, but I'm afraid if we add another childhood vaccine to the mix, and, it's bad enough that people who don't want to vaccinate their kids say we have too many vaccines, right? And here's another one.
I think that's part of the problem. I think part of the other problem is do you really have enough cases, to do an efficacy trial?
Right.
Maybe
so that's just a logistic.
Maybe
right?
for the respiratory disease,
right.
You could, not paralysis. Though.
Well, for the respiratory disease, we're kind of stuck, right? Because we're diagnosing it really rapidly clinically using BioFire, which can't discern EV-68 from rhinovirus.
Dickson, are you still there? No, I didn't.
Interesting.
I didn't think
we lost Dickson.
How did Dickson get lost?
He's not lost. He's contemplating.
I bet he was sleeping.
Nope.
Don't be mean.
I know. I'm sorry. I'm mean.
It wouldn't be mean if I was sleeping.
But I think I think if you just look at it, like there's more than 120 serotypes of rhinovirus.
Mm-hmm.
And
we did a paper a while ago showing you could make 100 valent rhino vaccine and get a response in mice. Mm-hmm. It was a while ago, but that was surprising that you could actually do that.
Wow.
But that's Marty Moore, my former student.
Oh, that's right. Yes, that's right. I remember that. Now, Amy, next year, 2020, there's next outbreak. Would be a perfect time to do a clinical trial of a D68 vaccine. Unfortunately, one will not be ready by then because you still have to go through preclinical and phase 1.
We're still discussing isolates.
But NIH is very interested in AFM and enteroviruses, right?
Yes.
There are a couple of meetings next year, and Fauci has written a paper saying we need animal models and we need more people to study this.
Yes.
To which we say, give us the money, right?
Which we say we've written several. Then we cry. And eat a quart of ice cream.
I'm interested. Where would you go for an animal model? What path do you think is appropriate to take for that? This is a tough problem.
So I have a path already. So one of the things is that there have been groups who've tried to develop animal models with cotton rats and ferrets. Unfortunately, the virus doesn't really replicate in those systems. They do develop an immune response, but none of those animals have ever been shown to develop the neurological consequences. And some of them have irrelevant, non-human physiological characteristics to their viral infection. So, for instance, the virus is found in stool from some of these animals. Well, we know that the virus falls apart in pH 5.5 or higher. And so it's really hard to understand how it's replicating in the gut to be shed in the stool. So probably ferrets and cotton rats are not the best animal model. So we're taking advantage of a system that's been developed that has huge genetic diversity in mice.
What's that called?
We're using the collaborative cross.
Oh, okay. All right. So this is a bunch of different mice that are very well defined genetically, but vary from each other in little bits. And you could screen those for a mouse that is susceptible to a D68 infection in the fashion that you'd like.
Yes.
Right.
Yes.
All right. That makes sense. That makes a lot of sense.
So, we've initiated. Yeah, we're waiting for mice to come out of quarantine to do this. But so we're hoping that since we already know that it's not ideal, but there is actually a gradient of infectivity in inbred mice, at the day one mouse model. So the suckling mouse models. So if you look at ICRs, BALB-C, C57, Swiss Webster, you see that there's an infectivity gradient. So ICRs are the most susceptible, whereas Swiss Websters are the least. And so using this very specific funnel cross between 5 inbred lines and 3 outbred lines, we feel we will be able to find a mouse that has all of those characteristics between the Swiss Webster and the ICRC. Whatever.
Okay.
I want to make a couple more comments. Yep. One on this paper. It warms my heart to see it loaded with one-step growth experiments that are assayed by plaque assay. It's just great stuff, and you can still get all of this information out of it. I also note that it has just 3 authors, not 4,000.
Yes.
Okay, that also warms my heart.
Yes, we're a small lab, and Audrey has done well.
What I want to ask is, when there's 3 authors and they all have the same affiliation, does it have to be superscripted?
It's a good question. I guess they wouldn't have to be, right?
I don't know.
Yeah. I don't know whether.
I don't know what the line. I don't know what
whether
the rules are.
you added the little A's or whether
did we
ASM added those.
No, I don't see any. I don't see any superscripts, at least on the online version that I've got.
So the little A's must have been added because we took them out.
Okay.
It was only so.
Didn't have it. We didn't have 3 A's. That's right.
Right. We took them out.
They put them in. It seems weird, doesn't it? Why would you?
Yes. Yeah.
I don't know. Why don't you call them? Ask them why their formatting is strange.
So just to wrap this up, how would you? It's basically in the title, but I'd like to hear your spin on how you would summarize the impact of this paper on the story.
So I think it addresses significant questions in the field that all EV-68 has the potential to infect cells of the central nervous system. Okay. Now, whether or not it has the ability to enter the central nervous system is really the pivotal question. And that's what we should be that's one thing we should be focusing on. The other thing it says is that what we thought was the receptor enhances viral infection, but it's not really required. And so we should take a step back. And I'm always asked this at job interviews about how much do we know about this virus? And when I say very little, they're flabbergasted because their gauge is what we knew about polio before we started to do research about polio. Mm-hmm. And we don't have any of that information. So we need to take a step back and say, we have all these modern techniques. They're great, but we need to do a lot of the basic biology about the virus using the old-fashioned techniques and answer those questions first.
So another thing that has come up throughout this discussion is the notion that acute flaccid myelitis could be the result either of a direct effect of virus on neurons or an indirect effect of the immune response. Can you elaborate on how that indirect effect would work, and are there examples of this?
So astrocytes are considered are almost analogous to macrophages. So
Okay.
PI at Stanford, before he passed away, kind of simplified it a bit, maybe slightly a bit too much, by designing A1 and A0 astrocytes. And so you can infect, so the idea is that you infect astrocytes, and these astrocytes become reactive. And they're also the major interferon secreting cell within the central nervous system. But they could secrete neurotoxins, and I'll get to that in a minute, that then kills the motor neurons. And we know that this occurs in ALS patients. So you can culture astrocytes from ALS patients with motor neurons, and those motor neurons will die from secreted factors by the astrocyte. And they have hypotheses about what those are. And I think those are like 263 neurotoxins. Wasn't that what the paper?
Secreted proteins.
Yeah, secreted proteins. So that would be indirect. We also know from viral infections, such as the related virus of Enterovirus, but also from West Nile and chikungunya, that infect both astrocytes and neurons, that if you are able to infect neurons, you get a very mild disease, or not very mild disease. You get a milder disease than if you infect both astrocytes and neurons. So one can imagine that if you infect EV-68 infects both astrocytes and neurons, it's more like 71 in chikungunya and West Nile. Or that if you infect astrocytes, that the secretion of neurotoxins is part of the immune response. And so you kill the neurons and you cause a paralysis.
How about more long distance? Like, could an infection in the lung cause CNS damage?
Like if you secreted cytokines?
Yeah. And are there examples of that?
It's possible.
Someone asked me that at a seminar once.
Yes, just recently. I guess, isn't that sort of like what occurs somewhat, very infrequently for flu?
Yeah. Yeah, there are long-range cytokine effects in flu, yeah.
It's possible, but flu also damages the epithelial lining of the respiratory tract. And it's not clear that EV-68 does. In some people's hands it does, in other people... so in like the cotton rat model, you don't really see any damage. Mm-hmm. It's not been investigated in the mouse, but in the human ALI culture system that a group in Switzerland looked at, they see damage.
So, Amy, when you got your own lab, what are you going to do?
I'm focusing on the establishment of an animal model initially. Okay. And then once my lab is established, then I'm going to come back to some of these basic fundamental questions.
Part of this must have to do with, from this discussion, it seems to me that the receptor is still something of an open question, right?
Yes, it's still an open question. And that's, so if I had my lab and I had two graduate students, one graduate student would do the mouse and one graduate student would help me identify the receptor. And then the third graduate student would do some of the elegant biophysics that needs to be done to understand the particle, the receptor interaction. So, for instance, Ian Lipkin's peptide is at the C-terminus of VP1. That should sit in the bottom of the canyon. So whether or not it's really exposed, if it's exposed during breathing, will it find an antibody? Is it blocked from exposure by receptor interaction? All of those questions are really unknown. And unfortunately, a lot of focus isn't really on the dynamics or thermodynamics of particle receptor interaction. So I'd like to go back and look at some of those.
Cool.
Good luck.
Hopefully.
Yeah.
Well, one day, hopefully, I'll have a job. Then I can talk to you from my job.
Yeah, we could.
Send the headset.
Send the headset. All right. Let's do some picks. Amy, what do you have for us?
So I have two. So we have an Enterovirus net website that keeps track of what we do. But there's also an Enterovirus in AFM, an acute flaccid myelitis organization or association now website that talks about the children and how they've been affected and the parents and the families and their support. And so they're able to merge with the transverse myelitis network, and some of the children go to Kentucky in the summer for day camp and stuff. So I think it makes it so that they don't feel that they're not within the mainstream of children. Yeah. That they're debilitated, that they're just regular, normal children, running up and down, trick-or-treating.
They may have a snip, though. But you wouldn't tell from just looking at them.
You don't want them to you don't want them to not be normal or not be what we've considered.
There's no normal genotype.
Right. But we don't want them to be
stigmatized.
Right.
Yeah.
You want them, if everybody is able to dress up as Halloween, go trick-or-treating, snow or no snow, you want them to be able to do the same thing.
This is good. This is what you can do in the internet era. If we had internet and polio days, we would have had the same thing.
Right. So I think making this more humanized and giving the children, so they don't feel left out, a more
cool. Very cool.
Normal lifestyle. I think it's good.
Rich,
on the Enterovirus site, you can update the October 23rd, 2018 entry about the bioarchive publication of the infection of nerve cells.
It's on the list.
We have to do that.
I came yesterday, but we got discouraged after Ayacook.
And we had to do Ayacook yesterday.
3.
Yeah, it was terrible. So it's one of my least favorite things.
Yeah.
Well, they always send it back. We never get the math right. We're defective.
Kathy, I have written for 35 years. You justify why you use the species. Mice are a convenient animal model, and this week they said that's no longer acceptable to say they're convenient. So I put in genetically malleable, but I've used that word. I've used that word for 35 years. Why this week is it not acceptable? Maybe they didn't read it before.
You got a different person reviewing it.
Hey, I'm just glad they didn't say you really don't know how to use the calculator. We got the 909 mice right. Let's just...
we often get the numbers wrong. Rich, what do you have for us? I can guess why we're doing
my pick is Tesla. We just recently acquired a Model 3 Tesla that we've been driving around, and I'm in love. But I want to advocate, because of this experience, electric vehicles in general, and I don't want to bash anybody else's electric vehicle, but I have to say that the Tesla is like driving reimagined. A friend of mine described it as an iPad on wheels. Which is pretty close to the truth. It's like they started with an electric motor and an iPad and a battery and built a car around it, rather than starting with a car and putting an electric engine in it. And it's amazing. And we have 2 cars, but we seldom use 2 simultaneously. So now we preferentially use the electric vehicle, and we don't go to the gas station anymore. It's amazing. It's just amazing.
Did you get a plug in your garage?
Got a plug in my garage. Put in a 240 volt, 50 amp circuit that connects directly to the main breaker panel through the wall in the garage, and you just bring it home and plug it in every night.
How long did it take from when you decided you wanted to get it till when you got it?
3 weeks.
Yeah, they're much faster.
So you didn't. Okay, you didn't have to put in and deposit and wait a year or anything like that.
No, but it actually, that's another thing, is that the whole purchase process is reimagined as well. You go to a Tesla, I'll say dealer in quotes, it's a showroom is what it is. And there'll be a couple of employees, and they're all salaried. The price for the car is the price you pay. It's not negotiable. Okay. And all of the they aren't salesmen, they're representatives, okay? And they're all salaried. So I hate buying a car because the fundamental assumption is that everybody's lying, okay?
Right. That's right.
About what they will pay or what they will accept. And that's all gone in this. These people are just there to introduce you to the car. And then you go home and you order it online. And the whole process, building the car and ordering it and everything else is done online. And you get emails and stuff, and you do pay a $2,500 deposit, but the car shows up. I mean, we're in Austin, Texas. The car came from California. They effectively built it for us, okay, and it showed up 3 weeks later, and we went and got a little orientation and then drove it off.
Cool.
It's amazing.
Most important question, what color is it?
It's a metallic gray.
Nice. That's my color.
All right.
Yep.
Yeah.
I like gray.
But they don't have the stick. You won't buy a car without a shift.
I know. But
this has never got.
Well, actually, I have the same. We had the same problem because my wife has a BMW. That's a stick shift, and Vincent, we've talked about this before about our love of stick shifts and how hard they are to find now. And this going to the electric vehicle from the stick shift Beamer is like it's a paradigm shift for us. Okay. But the Tesla has enough of its own gee whiz that you can kind of almost forget about the stick shift. And have you ever been? I think all electric vehicles are like this. I don't know that the Tesla is any different in this sense. Have you ever been in an electric vehicle and somebody punches it?
Yeah.
The acceleration is incredible. It's incredible.
Yep.
It's amazing.
Doesn't have
they
the
now
noise.
have electric race car races.
Yeah,
they're totally off the map in terms of.
So, Rich, have you played podcasts on that big screen yet?
No, I have not.
I need a picture of TWiV playing on it. Okay.
Okay, I will do that. It does have. It has a bunch of apps. If you're parked, you can now play Netflix on the screen. But it's got this one app that's a whoopee cushion. Okay.
So, Rich, all the electric refueling stations that Tesla has constructed, those are all free still?
I don't think they're free.
They used to.
We have not had the need for any of those yet. But that's amazing because you drive up to one of these Tesla stations, and the way this works is you just you plug it in, and it knows. I mean, you have a Tesla account out there in the cloud, and the Tesla station identifies your car and charges your account. Okay, so
it's cool.
It's amazing.
Enjoy.
This is where we're going.
He's going to die in his Beamer.
No, don't say that.
You're never going to get rid of it. You love the car.
I know, but that's not good to say that.
Well, you know how I meant it.
Dickson, what do you have for us?
Well, you know me, Vincent. I've got large optic lobes, so I appreciate visuals. And so this year's winners for the Nikon Small World Contest were announced, and they showed the first 20.
Wow.
Starting with the prize winners and working down to the honorable mentions. And it's just stunning. Every time I see them, I'm just blown away. Like Rich is in love with his Tesla. I'm in love with this. The number of variations on a theme, basically. That's what Nikon Small World looks like to me. And they get more inventive and more in depth in terms of quality of photographs and the mixed use of fluorochromes. And I just love it. I'm just in love with it. It's just, how could you not fall in love with biology once you see this?
It's incredible.
Yeah, it's nice when you
Amazing
open
stuff.
up and it has the top 20 all displayed.
Yeah, exactly.
Mhm.
Beautiful.
I love the spiders, man. They're just amazing.
Yeah, the small white-haired spider
Oh
is
my
amazing.
gosh. And the stentor are cool too.
Yeah.
With all
Yes.
I guess those are some kind of cytoskeletal elements.
Protozoans. Yeah, they're protozoans.
Nice.
I'm
Just great.
They're not. Stentors are single-cell organisms. Take a look at them.
They're single cell, yeah?
Single cell.
Wow.
I'm surprised, Dickson, that you didn't want to talk about West Nile being 20 years old.
Okay, West Nile virus is 20 years old too.
Yeah. There's a whole big review about it today in Plus Path.
Well, maybe that's my next week's pick.
Oh.
Kathy, what do you have?
I picked two papers with pretty similar titles. One is Unplugging the Pipeline, a call for term limits in academic medicine. The other is academic medicine ready for term limits? And the first one is by three people, two of whom are at Michigan, Whitney Beeler and Reshma Yagsi, and the middle author is Christina Mangurian. And basically, NIH has announced a plan to enact 12-year term limits for its tenured intramural lab and branch chiefs. And this second paper is advocating something along the same lines, recommending 2 4-year renewable terms for people in department heads, deans, and so forth. And then for educational positions like clerkship directors, they're thinking of 2 by 5 years. Evidently, Mayo Clinic has already gone to 8-year terms, and University of British Columbia 2 5-year terms. And the gist of it is that if things continue on at the rate they are, with department chairs and med school deans increasing slightly year by year in terms of the percentage of women, by the year, basically 50 years from now, it'll be 50-50. And so they say that's when all of this year, or, the current medical student crowd will have already retired. So that fact alone was just amazing. And so they talk about the pros and cons. And, the main con is that you might lose the institutional memory or experience that you get with, one person being in a position for a long time. But what you gain is new viewpoints, new diversity, new enthusiasm, and they discuss some of the things where, better decisions are made with diverse groups and so forth. And so it's an interesting call, and the fact that the NIH has done it, I'm wondering if it'll lead to some more academic medical departments doing it. And they also talk about, the second paper talks about how in politics, term limits have been found to be maybe not that effective in terms of getting diversity into state legislatures or so forth, but how politics is different, and maybe a better model is business, where there's a lot of evidence in business that if you get diverse groups, you make better decisions and your company makes more money. So just some things to look at.
I was going to say I want to have term limits in politics, because we are at the point now where politicians do not work for the people, they work for their party.
Just drives me.
It's true.
Right? I mean, they just want to get reelected.
That's right.
And I think
It's always been the
that
case.
is toxic, and we should have 1 term or 2 terms maximum.
You can't
Well,
have
it
one-
turns out in Michigan, they went to term limits. And so what happens is that, people are in the state house, and then they graduate to the state Senate. And this one article even talks about people kind of leaving early and moving on to the next one. And then you can end up having lobbyists write the bills instead of experienced people. So there's that danger in politics.
So the pinnacle is being a lobbyist. That's the end game, and there's
Right.
no term limits on that.
No.
Right.
No.
Well, you see a lot of senators and House representatives who've retired to become lobbyists. Like,
Oh yeah,
Yeah.
It's
I mean,
really,
the problem is, at least from one perspective, is it takes you so long to figure out what you do there, right? Isn't that like the saying about like the president, by the time your first term is over, you finally figured out how to be president, and then maybe you have 2 years of your second term and then you're a lame duck. When are you going to get anything done?
Yeah. So anyway, bringing it back to these papers, I like the idea that if we don't do something 50 years from now, this year's class of medical students will be retired and we'll finally be at 50-50 in terms of.
Yeah.
I'm all for academic leadership turnaround. Yeah, I'll go for that. I think chairs should be limited to.
Mm-hmm. Yeah, they talk about pretty much everything: chairs, deans, presidents. Yeah.
But is it really about the diversity, or is it also about the intellectual?
It comes with it.
Yeah,
right. Like turnover. Like you have a new set of ideas or of perspective of how to solve the problem.
Yep.
Right.
All right. My pick is a video about the coy wolf. Okay, why am I putting this up? Because we have these coyotes wandering around my suburban New Jersey town, right? All of a sudden they're showing up. And it turns out they're not just any old coyote, but they're wolf-coyote hybrids. And this is a video from Animal Planet that explains about these animals. I think they originated in Nova Scotia, and they have slowly been moving down throughout the Northeast. And the amazing thing, they live among people in highly populated areas. They take advantage of all the niches. Many of them move along railroad tracks, right next to people, and they move around at night, so you never see them. But they have different morphological features from either wolves or coyotes. They're very efficient killers of like raccoons and squirrels, and they eat roadkill all the time, deer. The other day there was a deer killed in front of my house, and the next morning it was eviscerated. It was probably one of these coy wolves. And I had no idea that this was a hybrid. So, and they have people studying this. They have an interview on this video with a biologist at Ohio State who has a grant to go out and they bleed them and they do genetic analysis. And, they look for pups in nests and they bleed them and so forth. Very interesting stuff.
So what is their natural predator that's going to keep the population under control?
So that's an interesting question. So they're often shot because people are scared of them.
That's a natural predator.
So they don't have one, and they are not afraid of people either. Normally, coyotes are very wary of people, but these are not. And in fact, our neighbor said one was running at her, and she got into her car right away. So,
wow, very
interesting. Coy wolf.
Going back to my pick, I forgot to mention that the one that's in the New England Journal, neither one of them is open access, but the New England Journal one, you can do a 3 free articles per month deal if you'll add your email to their database. So that's a possibility if you want to read it. It's short papers.
We also have a listener pick from an anonymous listener. Hi, Dr. R and friends. I saw this on my friend's Instagram as she and her daughter modeled this particular line of clothing in Michigan. I thought I should send it to you. It's great. And this is a line of clothing at Spill the Beans, etc. And it's shirts that say vaccinated, different vaccinated design. Hug me, I'm vaccinated.
These are wonderful.
I'm part of the herd. Vaccinate. I only date vaccinated boys.
Yeah, vaccinated because my mom leaves the research to the professionals.
These are really good.
Yeah.
They're very good.
Dr. Racaniello, you were my professor, and I still continue learning from you and your friends by listening to your podcasts often. I appreciate everything you and your team do. Well, thank you, Anonymous. Very nice. All right, TWiV 572, the show notes, links to all these things, microbe.tv/TWiV. Questions and comments, TWiV@microbe.tv. If you like what we do, consider supporting us. You can go to microbe.tv/contribute, and you could use PayPal or Patreon. Give us $1 a month or as much as you'd like, and it helps us to pay our expenses. We'd appreciate it. Our guest today has been Amy Rosenfeld on Twitter, ABR22. Thanks, Amy.
Thank you.
Good luck.
Thank you.
Dickson Despommier can be found at thelivingriver.net or .com. Is it .com? Yeah. Yes. And parasiteswithoutborders.com. Thank you, Dickson.
You're welcome. It was a good time, even from New Jersey.
I think when you're here, you're a little more loquacious.
Well, Amy and I talk all the time about this subject because I sit in the outer office, as you know.
Is that right?
So I actually had no questions for her because I've asked them all already.
Dickson, I haven't seen you in ages.
This is true.
I'm not sure. Well, I'm not sure I would recognize Dickson. You're still the gray-haired man, right? That comes.
I am. Here. Yeah. No, I'm still the gray-haired man. That's me.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
We were supposed to debrief about our Galveston trip. We'll have to do that another time.
Yeah.
It was a lot of fun.
Yeah, it was a lot of fun.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by Vincent Racaniello. I shouldn't refer to myself in the third person. Should I say me, right?
Yeah.
Well, you could do third. I think third person is effective.
Is it?
Yeah.
Okay. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.